Good afternoon, Mindsetters, and welcome to yet another beautiful spring day edition of Mindset Learn Extra. My name is Kateho. It is, of course, proudly brought to you by the Department of Basic Education. We are going to be together for an entire hour doing nothing but paper 2 of your question papers in your exams. And I'm not alone in the studio, as you guys might know by now. I am with Cheryl. How are you?
Afternoon, Kateho. Well, and you?
I am awesome. What are we doing today?
Welcome, grade 12s. I know you're not used to this face. You're used to Aslam, but unfortunately, he can't be here today, so I'm going to be standing in for him. We're looking at DNA and genetics, especially, orientated for paper number 2. Yes.
Awesome. Just stay tuned. Of course, we're going to be doing all of those questions that you guys have. That might be a little bit challenging or complicated for you, but then we're going to be doing them together with Cheryl here. Just go make sure that you go to our Facebook page. It's at www.facebook. learnextra.co.za. Just go there and then you throw your questions and we'll make sure that we attend to all of them. And then just make sure that, of course, you get in touch with us on Twitter as well. We are at Learn Extra with an X. Just go there and throw all of those questions that you might reckon that they are difficult or challenging to you as a grade 12 learner. It's paper 2 of Physical Sciences and it's Spring School Edition, proudly brought to you by the Department of Basic Education. I am Kateho and I'm going to be throwing it to Cheryl to take it away.
Thank you. Sure. Speak so fast. I'm a little bit gobsmacked there. Welcome, grade 12s. Welcome to the Spring School Edition. I think by now, all of you, I know all my grade 12s are so tired and they're so, we don't want to do anything. But it is almost there. The end is in sight. You guys have, I know the big finals come up. You've gone through the prelims, all right? And now's the time. If you haven't buckled down, all right, you can do it. The end is in store. It's 75%. You guys just give it all your best, and I know that you're going to be able to get through it. What we're looking at today is paper 2. One thing I do want to stress, please, and I think my kids are also tired. I have 3 girls that I give extra lessons to as well, and all I do is give them past papers, and I can see their eyes are rolling and going, oh, not again. It is the most important way to revise for your exam. The information is available to you on the Mindset Network, on the Department of Education. Just look for past exam papers. You can go as far back as 2008, all right, if I'm not mistaken. Use the kind of questions that they ask. It is such an invaluable task. Okay, so we're looking at paper 2 today. We're looking specifically on questions on DNA and genetics. So let's sit back and let's get down to work. Okay. The multiple choice questions that Aslam has chosen today for you are quite tricky, all right? They involve quite a bit of reading, and don't skim through it. In the beginning, when I started going over it again, going through the answers, I realized, sometimes I had to go back again because I didn't read the question properly. Okay, so when it comes to it, lots of information coming at you. Let's take each piece bit by bit. Okay, we're going to start with question number 1. All right, if I can get it going. Okay, it says here, which one of the following serves as evidence of cultural, evolution in early Homo sapiens? That is a very key word when we get to cultural. It's talking about, their lifestyle, all about their lifestyle. And if we have a look at the options that we're given, we are looking at drawings and carvings on rock, animal remains, male and female skeleton, more than one Homo skeleton. Each of them, when we look at them, reply to the skeletal remains. The skeletal remains is paleontology. Doesn't tell us about what they used to do, It tells us what they look at. So the answer, for number 1 is going to be A. Cultural, your drawings and your culture and your carvings on the rock tell you about the interaction that they had, the people had at the time, okay? Question number 2. As I said, we're looking at DNA and genetics. Question number 2 says here, a father has blood type A. Very important, all right? When we get to blood types, remember, he can be, heterozygous, or he can be homozygous. Very important. He has 4 children with the following blood types. Now, one child has A, one child has O, one has AB, and child 4 has number B. Okay, now from that information, they want to ask you, what is the blood type of the mother, all right, of all of the children? Now you need to be very careful. When it comes to the blood types, this one over here, O, is always going to be the important defining blood group, because when you have an O, both the parents must be carrying, all right, both of them. It's a recessive trait. It's a recessive trait. So both the parents must be carrying, all right, the O. If this child wasn't, then we could have, it might have been plain, much more plain cut, all right? But this child, as soon as you see the O, now you say, oh, what both parents must have the O for this child to be O. So if daddy then was A, all right, there we go, there's your A's, but we need to have a B, all right. So what must mom have been there? Mom could not have been AB. Oopsie, sorry. That is the one you scratch out immediately. Codominance, all right? When we have codominance, we won't have the O, right? So what did we have? If daddy was A, and to get the B and the other children, definitely mommy must have had B. So if we were to write it, she would be BO, and he would be AO. All right, and from that you can see one of the children, AB, one of the children would have been B, one of the children A, and one of the children O. So if you do your cross there, you would have seen, all right, how you got that. That O as your choice, all right, is a very important hint. Okay, next question. Lots of words going through. Remember, when it gets to genetics, and we're talking about, all right, the evolution and those things as well. There's a lot of terms in there that I'm going to be honest with you, it's difficult to try and explain. All right. So your terminology, you need to be able to know your terms. Okay. So question number 3, new alleles. Remember, what are alleles? Gene expressing a characteristic like brown hair, all right, tall, short, in a sexually reproducing population. All right, new alleles arise. How do we get new combinations? That is the most important. How do we get new combinations? Now, if you have a look at the questions, right, it seems like some of them can be correct. Number A, mutations in DNA sequencing prior. Prior means before. All right, before meiosis. All right, keep that question in the back of your mind. Random fertilization of gametes during reproduction? No. Why do I say no? Random fertilization, all right, just simply means what has already been formed. The sperm has already been formed, and the egg cell has already been formed. It's not going to give you different alleles. The alleles are already formed. All right, it's the combinations of already formed ones that are going to be there. So it's not number C. Number C, random assortment during meiosis? No. Have a look. This was such an important word. The prior. The prior. All right, where must everything happen? How do we get new alleles? Exchange of chromosome segments between homologous. What do all of these give us? What do all of these give us? They give us variation, yes, all right, but they don't give us new ones. How do we get new, all right, alleles? We get them by mutations, all right. Mutations cause change, bring about something new. Okay, question 1.4. A tall pea plant was crossed repeatedly with a short pea plant. In each of these crosses, they produced only tall offspring, okay? So tall was crossed repeatedly with a short. In each of these crosses, they were all tall. So what do you think I could have put here? A big T, short would have been a little t. So what can we see? That tall is probably dominant. Okay, now let's see. We can conclude that tall pea plants involved in the cross is homozygous. Is that true? In other words, are they all big T, big T? Okay. Or number B, tall pea plants involved in the cross is heterozygous. Okay, so we've got homozygous. Offspring are all heterozygous for height. And short pea plant involved in the cross is heterozygous. So we know this one is definitely out. Because what do we know about shorts? If it's the recessive is always homozygous. So now we're left with A, B, C. Okay. The tall pea plant involved in the cross is homozygous. Tall pea plant involved in the cross is heterozygous. Now you should have tweaked. If I have two heterozygous plants, remember my laws, all right? What can happen? 25% chance of them being short. What did it say here? Only. So each time I cross them, only were tall, all right? Number C, offspring are all heterozygous for height. Okay? Do you think that's possible? Not necessarily. What can we conclude? Tall pea plant involved in the cross is homozygous. It must be. What is Mendel's first law of dominance? All right. When we cross a dominant homozygous with a recessive trait, the dominant gene will always, all right, be the one expressed. Okay, so that's going to be question number A. This is a very tricky, this was quite a tricky one. It says the table below shows a section of mitochondrial DNA. Do you see how we can ask DNA, all right, not necessarily by itself, how it comes into the, all right, our evolution section as well. The sequence for a modern human here, a chimpanzee, and 3 hominid species, all right? So we have modern, chimpanzee, and hominid. Now this is the most important part. The letter X in the chimpanzee and the hominid sequences means that the DNA basis was the same, NB, 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 as that found in the modern human sequence. In other words, where we see an X, that means they all share, right? They share a common characteristic. Now, oopsie. Okay, now the next question is, oh, and it's disappeared. All right, let's have a look over here, sorry. Okay, for the table, we're going to come back to that one just now because all the different information there Murphy has done its thing. Okay, question number 1.6. The following data represents a small section, okay, a small section of a sequence of nucleic acid bases taken from an animal cell, all right? And there they are, A, G, C, U, G, or C, G, U, U. Okay, so nucleic acid, we know it can only be one of two things, DNA or RNA. Okay, now let's read. Now this one was tricky. From this data, it is reasonable to conclude, okay, that number A, this portion of nucleic acids will code for a chain of 8 amino acids. 1, 2, 3, 4, 5, 6, 7, 8. How many of you are automatically going, yes, 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 that's the one? 8 bases, 8 nucleic acids. Let's go back to translation. All right. My bases are paired in groups of 3. Okay. Not 8 amino acids. There we go. A group of 3. That would be an amino acid. And that would be an amino acid. And we haven't even finished there. So you need to be careful. When I read that one first, I was thinking, oh, that could almost be correct. And I read 8 amino acids. Right? Not 8 complementary bases, 8 amino acids. And one amino acid needs how many bases? 3. All right. So number A is definitely not correct. Number B. The sequence given will be complementary. All right, here when we talk about complementary, we're not talking about opposite, right, but it's their pairs. So the sequence given, all right, what will their complementary bases be? C goes with A. Does C go with A? Does G go with T? Already I can see. No, they're not the complementary bases. So number B is incorrect. All right, question number C. The nucleic acid shown contains the sugar ribose. What is ribose? Which sugar? Ribose is found in RNA. Is this an RNA molecule? Yes, it is. Why? What does RNA have? Uracil. If uracil is present, I can conclude, all right, that the nucleic acid shown contains ribose. Right, let's look at the last question. The nucleic acid shown is DNA. What did I have in it? A U. Do I find uracil in DNA? No. All right, uracil is only present in RNA. So, definitely wrong. Therefore, I know that my answer is going to be C. Okay, question number 7. Okay, we have 4 sequences in which the replication of DNA. Okay? We have the list below provides information relating to replication of DNA. What is replication of DNA? 1 strand of DNA, and what do I have at the end? 2 identical strands. Okay, so now we're given the order, 4 steps. Complementary nucleotides bind to each other. Sugar phosphate bonds form between the nucleotides. The newly formed DNA molecules are identical to each other. Number 4. After unwinding, the DNA molecule forms 2 single strands. We're going to have a quick break, I believe, Kadleho. Time flies when you're trying to explain everything. Okay.
Indeed, Cheryl. Of course, I must remind you guys that this is our spring school edition of Mindset Learn Extra, proudly brought to you by the Department of Basic Education. Do tune in with us. When we come back, I'm going to be telling you about the exciting competition that we are running. And of course, I'm going to be giving you links to the questions that you guys might have. It's Katleho and it's Mindset Learn Extra. We'll be back in a moment. Welcome back, Mindsetters. I am Katleho, and of course, you're still tuned in to our spring school edition of Mindset Learn Extra, proudly brought to you by the Department of Basic Education. As usual, I'm not alone. I am with Cheryl, and we are doing paper 2 of your of your life sciences today. Remember to throw all of those questions that you guys might have, challenging questions, challenging or not, to our Facebook page, and we'll make sure that we attend to them. I must read you something though. Here's paper 2, life sciences challenge. See how many of these questions you can answer. You must create a a free account, and then enter the Curio code, which is A6FA0. It's A6FA0, and then you'll get immediate feedback after submitting that. And I have an interesting question that I'm going to be posting to you guys a little bit later. I'm in competition rather. And then just do tune in with us, and then we'll make sure that we attend to all of those questions that you guys have lined up for us on our Facebook page. Cheryl.
Oh, he talks so fast. I'm like, oh, how does it? I tell you, the older you get, the slower the brain goes. All right, guys, I know you're getting used to my back here, but it's just much easier for me to explain on the board here, and my back is a bit of my better side. All right. Okay, if we have a look, what we were looking at, the question was, all right, there's information relating to the replication of DNA. In other words, taking the DNA molecule and we're making two identical strides. Now, the question wants you to put them in the correct order. Okay, so we need to put from number 1, 2, 3, 4 as the whole process occurs. Okay, so let's start off. Number 1, already the complementary nucleotides are binding to each other. That we can't start off there yet. Number 2, the sugar phosphate bonds are also bonding. All right. Number 3, we've got this whole new DNA. So we need to start at the beginning. And where does it start? Look at number 4. It says after unwinding. Right? What does the DNA helix have to do? It needs to unwind. All right, so we have our 2 separate strands. So now we're looking for a question. Number 4 is the first phase. So already, if you are clever, right, we know that number A and number B are incorrect. Okay, now what happens? We have these 2 single strands. And what has to happen? Now the complementary bases are going to go to each strand. So now let's look. Which one was that? Now the aha. All right. We're looking for number 1. Now, do I have to look further? Yes, if you want to. But I already can see 4 and 1. Okay? So what is happening? Question number D is the correct sequence. So I unwind. Complementary bases are going to form, but I need to have like a stopper. What do I need? All right, and the sugar and the phosphate need to form, all right, the rungs and the base of my ladder. And what do I end up in the end? Last one, question number 3. Oh, excuse me. Number 3, 2 newly formed DNA molecules that are identical to each other. Okay, another quick, tricky one. They're always tricky for me, because remember, you guys, always remember, because it's multiple choice, that the answers are given to you. You have this feeling that it's easy. Read your questions through very carefully. Just skimming through them, you could just miss something vital, just a small little point that could make the difference. And it's 2 marks, all right? It's 2 marks in your paper, and actually can make a big difference. Which one of the following accounts for gametes? All right, what are my gametes? Those are my sex cells. In other words, my egg and my sperm, if we're looking at humans, have a single allele, or allele, whichever way you want to pronounce it, have a single characteristic. Remember, all right, when we have a look, 46 chromosomes, how many alleles did you have? You had 1 from your mom, 1 from your dad, so you had to have 2. All right? During meiosis, what did we do? We divided it so that 1 characteristic can go one way and 1 the other way. Okay, so now, which process is going to ensure that my gamete, my sex cell, only has 1 characteristic? All right, instead of 2. Now, again, you look at question A, and the first thing you get, yes, because it says here, the chromosome number is halved. And that's where you stop reading. No. All right, don't stop reading there, because it says during meiosis 2. No. Where does the chromosome number halved? Meiosis 1. So the statement is wrong. Number A, gone. Sorry for you. Number 2, Mendel's principle of segregation. Aha, segregation. What happens here? Aren't they 1 to one side, 1 to the other? Isn't that segregation? All right, when we take the 2, segregation means to separate. 1, 1. Okay, so you should notice by now when I put a question mark, it's usually the right answer. Number C, Mendel's principle of independent assortment. Right, independent assortment just means that they're randomly chosen. It doesn't say to you specifically, all right, that they each go to their one half. And the law of dominance, no. All right, that has not got anything to do. It's just when you combine your alleys. So the answer is number B, segregation. Because what do? We take two and we segregate. We separate so that we've got one in one and one in the other. Okay. Question number 9. In multiple means many. Right? In many allows ... How are we going to finish that sentence? More than one gene controls a trait or characteristic. All right, more than one gene. Let's think. Multiple does have many. All right, think about it. Let's go. There are more than 2 different alleles for the same gene. What does multiple mean? More than 1, more than 2. So in a multiple alleles, sorry, there are more than 2 different alleles for the same gene. Also can be a possibility. Number C, the different alleles for the same characteristic are at different positions. No. When we talk about positions, that's going to be locus. Okay, there are only 2 alleles for, not only. Multiple means many, not only 2. So, let's go back. Eeny, meeny, miny, mo. In multiple alleles, more than one gene controls a trait. No, it is number B. There are more than 2 different alleles. Multiple means more than 2. Okay, our last multiple choice question. In a situation where a characteristic is expressed, all right, expressed, I'm going to see here for you, seen, all right, or shown more frequently in males. Okay, that sentence should be bringing off alarm bells. What does the sentence say? Characteristic where more males get the condition than females. What's the alarm bells going off? What's the alarm go? All right. What one should you be thinking of? Sex-linked trait. Why? That little y, right? Men get it more often, expressed more frequently in males than females. So, what is our options over here? One allele is dominant over the other. That's going to happen, yes, but it doesn't account for why does it happen in males than in females. That's just random. The alleles for the characteristics are located on the X chromosome. Who is the carrier? The female is the carrier. Where is the gene expressed? In the male. Okay, notice, question mark. Let's carry on. The alleles for the characteristics are located on autosomes. Wrong. Autosomes are your body. You're right, you want gonosomes. Gonosomes are your sex chromosomes. So that is out. The alleles for the characteristics are located on the Y. No. Look how small the Y is. This Y is so small it can't carry anything. All right. So it's because the female is the carrier and the male. All right. Not. Okay. Next thing we're going to go to is our terminology question. A few of them I actually have made mention of in our multiple choice questions. Okay. We'll go through these a bit quicker. An allele that does not influence the phenotype. Phenotype is what we see. When will it be hidden? What part is hidden? Okay, oopsie, there we go. Oh, it's not opening. Okay, I'm going to just press over here. Okay, not opening, so I'm going to write it. The allele that does not influence the phenotype is called the recessive. All right, the recessive trait. Number 2, the position of a gene on a chromosome is a locus. Right, number 3, the physical and functional expression of a gene. Physical, what we see, all right, and what it does. That is the phenotype. Right, as soon as we see physical, physical is what we see, functional we can see, all right? What is the one that we can't see? That is what's in your genes, and that is your genotype. Okay, question number 4. Chromosomes that are not responsible for sex determination, all right, that is going to be your, all right, autosomes. Did you see in the multiple choice question we used it? Not responsible for sex determination. That's the key word. Remember, your X and your Y, what are they called? I wrote it perfectly. Your gonosomes, your gonads, your sexual reproductive organs. Okay, and the last question, question 5. The process of finding a desirable gene, isolating it, and then moving it into cells. All right? It's not cloning. Cloning is making an identical copy. Okay? Here, we're manipulating. We're taking what information we want and we're changing it. Okay? There's 2 terms that you could have had over there. One is genetic engineering, or another one you could have used is DNA manipulation. There we go. All right. Okay. So remember the format of your paper. Start off with multiple choice, then there's terminology, then we're going to the next section, which is usually your matching. Not so much matching columns. It is, all right, but it's the tougher version. Okay. For each of them, you have a statement, all right? You have a sentence. Then that's in column 1. Then in column 2, you are always given 2 choices. Now, you could answer 4 things. You could say that only A relates to that statement, or only B, or both of those statements are true, all right? Or we have none. None, all right? None. None of this statement, none of those in column 2, right, have got anything to do with that in column 1. Okay, so let's start. Okay, the statement is, discovered the double helical structure of DNA. Your 2 answers are Francis Crick and James Watson. They both did, all right? They both did. So your answer is A and B. Number 2, it describes an evolution as consisting of long phases of little change, then all of a sudden, a short phase of rapid change, all right? The answer there is A, punctuated equilibrium. What is punctuated equilibrium? All right, that things remain the same, and then what do we have? We have some change occurs over a quick period of time, and then we carry on. Darwinism has got nothing to do with that. All right, question number 3, variation within a population in which there is a range of intermediate phenotypes. So there's a large range. There's lots of different ones. Your options are discontinuous and continuous. It can't be number A, because what did it say to you? There's a large range, all right? There's lots of intermediate, so it means that it must be a continuous variation. It's continuing to change. It's continually bringing new sets of characteristics. Evidence for evolution, mitochondrial DNA or a cladogram. A cladogram doesn't show evidence for evolution. It just shows you evolutionary trends. Okay, so again, all right, that was number B, A only. 2 alleles of a gene that are equally dominant, all right? Complete dominance, no. Equally co-dominance, like our blood types. And our last one, bonds that hold amino acids together, protein peptide, right? Hydrogen bonds hold together in DNA. I think we're ready for a break, Katlego.
Of course.
I need water. Yeah.
Of course, do remember, guys at home, that we are doing paper 2 of life sciences today. It's very fascinating, DNA and genetics. Do tune in and keep those questions coming. I am checking my Facebook page, and those questions are trickling in. Me and Cheryl will attend to those in a moment. I must remind you guys, rather, that you must enter the Be a Bright Spark competition, where you stand a chance to win a Chevrolet Spark L. It's very, very easy. You just go there and download the Safeway K53 app from your Samsung Galaxy store, or buy that Safe manual book, and then you can stand a chance to win that beautiful, beautiful Chevrolet Spark L. I'm coming back after this competition. We're going to be more or less still attending to your questions. Keep them coming. We'll be back in a moment. Hello, Mindsetters, and welcome back to a beautiful spring school edition of Mindset Learn Extra, proudly brought to you by the Department of Basic Education. I am Katlego, and I'm with Cheryl, our teacher today. And before we move to question 4 of our Life Sciences Paper 2 questions, we are going to be attending to some of the questions that you guys threw onto our Facebook page. Remember to go to our Facebook page, guys, to throw all of those questions that you might need for us to attend to them. It's at www.facebook.com/learnextra with an X, and we'll attend to them. The first question is from Ndivu Wondou. He's saying that what is the difference between a homozygous and a heterozygous?
Okay. Remember, when we're looking at genetics, there are a few terms that are essentially important. Okay. Let me explain to you quickly. I'm going to use this. Let me just use this pen quickly. All right. When we're talking about homozygous and a heterozygous, here, all right, this is you, okay? That is you. Now, you received, let's take eye color, all right? You received, I'm going to put an eye here and an eye here. I'll erase it all just now. When you received your eye color, all right, from your mom and your dad, okay? So you received one information set from your dad and one information set from your mom. Okay, now, if your dad gave you brown eyes, a brown color, dominant, and your mom gave you also a brown color, that is homozygous. What do we mean when we talk about homo? All right, even when we use the word homosexual, etc., we're talking about 2 individuals that are the same. Okay, here, mom and dad gave you the same characteristic. That means homozygous. When we talk about hetero means, all right, maybe over here, dad gave you a brown for brown eyes, and mom gave you the characteristic for blue eyes. Are they the same? No. Look, a big B and a little b. So when I have 2 different characteristics, heterozygous. Okay, so homo, if I've got 2, I've got 2 of the same characteristics, or if I'm heterozygous, I have 1 of one and 1 of another. I have 2 different types.
So there you have it, guys. Homo same, hetero different. Just keep it in mind.
Got it.
We're going to be taking one last question before we move to question 4. Ntombi Butelezi is asking, is the capital letter allele always dominant to the
Always.
other?
Always, always. When we use capital letters, we are showing dominance, okay? With the question that I want you to stick to the question now, because you're going to do a question specifically on heterozygous and homozygous, where you're going to see this. We always use a capital letter to show dominance. A small letter is always used to show a recessive trait. It's secret genetic code. All right, capital letter, dominant, recessive, always in the lowercase. Okay, I noticed one of the questions you were asked there about where they can find life science papers.
Yes.
Tell them, go to the Mindset website
it is
and
there.
also the Department of Education. They really have got all of those papers with the memos. Awesome. The Department of Education gives you a lot of the past papers. That's where I go. All right. That's where I go. Okay, guys. Now the next one. Oh, let me quickly just erase all the I's off there. Okay. The next question that we're looking at is to do with, all right, a dihybrid cross. Now, a dihybrid cross, all right, is where we're going to cross more than one characteristic. But you don't have to do the whole cross, okay? They're just going to show you. You need to just read your questions, and I'm going to go through it with you, and we'll see, okay? It always seems like so much, but I like to write things down and simplify things. Maybe if you do it, it might be easier for you. Okay, let's have a look. It says here, in a pea plant, the allele for round seeds is R. Okay, now I'm going to rewrite it on the side. We know round is R, okay? And is dominant over wrinkled. Write it on the side. The reason being, because we can go back to it. We don't have to go through the whole thing. Okay, so what have we got? We've got two characteristics here, a round seed and a wrinkled seed. But what do we also have? The seed can also be yellow, and it tells you, even though it didn't tell you it's dominant, it tells you that by using the capital letter. Okay, so we have round seeds, which are dominant. We have wrinkled seeds, which are recessive. Okay, we have yellow seeds, which are dominant. Or we can have a green seed, all right, which is recessive. Now, that information, right, you already can start to plan your genotypes and your phenotypes, okay? When it comes to the dominant, that can sometimes be tricky because my dominant can always have two options. My dominant can always be, all right, big R, it is homozygous, or it can be big R, little r, heterozygous. My yellow as well. My yellow seed can be a purebred, homozygous, or it can be heterozygous. Okay. But this is the one thing about a recessive trait. Recessive traits are always homozygous. They're always two small letters. All right? So my wrinkled seeds are always going to be WW, and my green is always going to be, sorry, not WW, RR, and YY. Okay. We keep that to the side. Now they want us to do something. Plant number A. All right. Now look at the question. It's going to ask us, what is the genotype? Genotype is the genetic letters that I want, not the phenotype. Okay. Plant A is, number one, heterozygous for both seed shape, heterozygous, all right, for both seed shape and seed color, all right. Plant, okay, was crossed with plant B, which had wrinkled green seeds. Now let's start off there. Okay, plant A. Write down the genotype of plant A. Heterozygous. Let me go back here. For both seed shape. So, it was round. Let me go back to seed shape. It was round. All right, but what was it hiding? It was hiding, all right, the wrinkled. Think carefully. All right, and then what is it also? And seed color, heterozygous. Why? All right, okay, let's go and see behind door number 1. Have a look. Go to that one over there. Have a look here. Write down the genotype of plant A. It said it was heterozygous for both, all right? Which means, there we go. Let me get to my pen again, all right? There it is. There it is. It's telling you. It shows the dominant, but it is hiding the recessive. Now let's go to plant number B. Plant number B was wrinkled. What was wrinkled? Recessive. What color was it? Green. Green was recessive. All right. When something is the recessive trait, it can never be heterozygous. It always has to be, it always has to be homozygous. There we go. All right. They gave you all of the information. As I said to you, it's easier to write everything down on the side there so that you could keep going back. Now, write down, this is the more trickier, write down all the possible genotypes of the gametes of plant number A. Let's go back to plant number A. Here we go. All right. This is plant number A. When we have it was heterozygous, now they want to know what possible gametes can that plant number A, all right, what possible genotypes will it show? Now, if we have a look over here, I want to show you how we got to that answer. All right. Oopsie. Okay, I'm trying to get all the things here, and then it's going. Okay. All right, so here's our pencil. Have a look over here. Okay, we had R, R, Y, Y. Okay. Now, what do we know? We need, how many gametes need to come from that? 4. All right? And I might do it a different way to you, but what I do is I take one characteristic, all right, from each of them. So I have 1. All right. There we go. I have another one. See, RY, or little y. Then I take my big Y and I cross it with a little r. There we go, like that. And I take my little I and I cross it with a little r. All right, so if we were to do it again, let me show you. All right, maybe your teacher has shown you a different way in which to do it. All right, but I go, R goes to 1. So I have an RY. Then I have my R go to the next one, all right, which is big R, little y. Then I take my little r, and I go to big Y, and I take my little r, and I go to small y. This gives me, all right, my gametes. In other words, my sex cells. Now you can't have, remember what does a gamete have? It has 1 allele in each. It does. It might have 2, but they're 2 different things. So one letter codes for shape, all right, and the other letter codes for color. Okay. Now it says the next one. State the phenotype of an offspring having the genotype RRYY. What are we asking you? I'm asking you now to put that into words. Okay, what does RR mean? What does RR mean? But if I go up here, what does RR mean? All right, RR means wrinkled. RR means wrinkled. What does big Y mean? Let me go back here. Big Y means that it is yellow. So if I go, all right, it's wrinkled and yellow. My phenotype says, don't give me letters, all right? I'm not Cheryl, I'm Cheryl, all right? That's what my code means, okay? Don't the letters just show me the genes, but what am I showing, all right? That is the phenotype. Again, let's put that into words, all right? They are round, let me go, my pen, all right? They are round because it's dominant, and they are yellow. Again, what do I see? My dominant, all right? My dominant characteristics. Okay, again, 2 heterozygous. Big R, little r. Big Y, little y. Different heterozygous. Okay, now your plant B, right, let's go back here. Plant B, all right, was little r, y, y. You crossed with plant C, all right, and all the offspring had round yellow seeds. All of them had round yellow seeds, all right? Round is capital, yellow is capital, all right? It says, use this information and write down the genotype of this plant. Have a look. It said to you, all right, that you had a recessive plant. And when you cross them with this plant C, they didn't tell you about plant C. All they said to you about plant C is that what do you got? All the offspring showed dominant characteristics. Mendel's law of dominance. Homozygous dominant crossed with a homozygous recessive, what will you always see? The dominant trait. Are we ready for another break?
Of course, we're going to be
Belinda
taking
whispering in my ear here.
Oh, we're having so much fun doing life sciences paper too,
Listen,
guys.
yeah, if you say physical science again, I'm going to run screaming. It's the one subject that scares the living daylights out of me. It's like maths. Yes.
It's life
frightening.
sciences, guys. I must just remind you that you are tuned into Mindset Learn Extra. The spring school edition was proudly brought to you by the Department of Basic Education. We are going to be taking a quick break. When we come back, we'll be doing more and more of those questions that you guys might have for us. Stay tuned. Welcome back, everybody. You're still tuned into your beautiful Spring School edition of Mindset Learn Extra, proudly brought to you by the Department of Basic Education. They are behind us, guys. And of course, before we move to Cheryl, I just need to attend to a question by Tido Pamitungu, who asked, she's in grade 11, and then she just registered for that Curio questions, that tag that we have going on there. The question is, will I be able to log in using my account, even if you post other practice questions, question papers for other grades, even though I'm in grade 11? Never fear, Tito, we have you. You can still go there and play around with it and then answer as many questions as you can, and then you can just still participate even though you're in grade 11. It will be some sort of a preparation for when you get to grade 12 next year.
I must right out of my mouth. Well done, Katlego.
I must just remind you guys that the link is there. You create a free account, a free Curio account, and you enter the Curio code. It's A6FA0. A6FA0, and then you just go there, you answer as many competitions, or as many questions rather, as much as you can. And then I have a Enter Be a Bright Spark competition, where you stand yourself a chance to win a beautiful Chevrolet Spark L. You just go there to our link and download the Safeway K53 app from any Samsung Galaxy store, or buy the book, and then that you stand a chance to win this beautiful Chevrolet Spark L. But right now, I'm just going to throw it back to Cheryl.
Okay, the diagram behind me should look very familiar to you, all right? The question now, we're going to look at DNA synthesis, the translation, and transcription. Okay, the picture is on different sides than the questions, so don't get too confused. I'm going to go forward, back, forward, back, forward, back. We're going to be doing like a whole little dance now. All right. As soon as you see this question, all right, the diagram below shows the process of protein synthesis. Oh, we're not going to, I don't like white. All right, there we go. So the whole diagram in front of you, you should by now, you need to realize this is going to be a question that you're going to get in your paper, okay? The section on the protein synthesis, the translation, and the transcription. Okay, we're going to go through, all right, each of the questions. As I said, we're going to go chunk, 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 chunk. All right, now, it says use the diagram below, and that's the one on the other page. Identify the following. Question number 1, have a look. There is a difference between what a molecule is and an organelle. So if I look at molecule X, as you can see there, is going to be my DNA. All right, and the reason I know that, number 1, it's inside the nucleus. Number 2, it is double-stranded. Number 3, all right, it also is going to be in a helical shape, and that is why I know that it's going to be a DNA molecule. Question number 2 asked you for what organelle? All right, an organelle is a structure within a cell. Okay, a structure. Go back to grade 10. I know you all can't even remember what you did yesterday. Go back to the structure of the cell. The cell, excuse me, has got different little compartments, different organelles, right, that are responsible for certain structures. So, number Y, what is this organelle called? And by now, you should know it's a ribosome. So your 2 organelles here, your organelle is a ribosome, and this would be your nucleus. Your molecules are smaller, all right, than your organelle. So there we go, you'll see DNA and your ribosome. Now, I want you to identify the nitrogenous bases labeled 1 and 3. If you go on this diagram over here, you will see, all right, there's number 1, there's number 2, and there's number 3. You should recognize this as a tRNA, okay, and here the bases are C, okay, C, and an A. So number 1, what is the complementary base of C? It is always G. And number 3, what is the complementary base of A? It is on the RNA. So number 3 must be uracil. Okay, so let's go over here, you will see. Okay, number G, all right, and uracil. Now, the next question is important. This is where, when it comes to these longer questions, how you say something becomes important. All right, now it says there, what role does DNA play during transcription? What role, if we go back over here, use the picture. What role does DNA play? First of all, let's think. First thing, what function, what does DNA do? DNA actually codes for the protein. Okay? Now, why can't it do its function? Because it's too big. All right? It's too big. It can't go out the nucleus. So what does it do? All right? It takes one of the strands and it uses it as a template. All right? What does a template mean? It's got the message on the side already. Now, what do I just need to do? I need to make another code on a smaller strand so that it can move out of the nucleus. So my DNA codes for the protein. Okay, the DNA slightly unwinds. What does it do? All right, its one strand is the template strand. And what does it make? It makes this, a messenger RNA. Okay, so if we have to go, all right, let's see how, if you were to answer. DNA codes for the protein. So there we've got what it does. All right, DNA, but it cannot leave the nucleus. So we know DNA codes can't leave the nucleus, but what does it do? One of its strands is used as a template. And from that template, what do we form? mRNA. Okay, that is the role of DNA during transcription. Now if we go to the next question, it says, describe the part of protein synthesis shown as process W, which occurs at Y. Oops, wrong one. Okay, let's go here. Oh, never mind. Okay. They wanting you to tell me everything that's happening over here. All right. So you need to explain to me what is the most important thing. Let's start. Transcription that we know, all right, what's going to happen? tRNA, what does it pick up? It picks up an amino acid. All right, I always refer as tRNA as taxis, because that's exactly what they do. Don't they pick up passengers? Okay, so tRNA picks up, all right, its amino acid. And how does it do that? It all depends on its anticodon. All right, this base over here will determine what gets picked up over here. Now, what does a taxi do? It takes people to places. So what does the tRNA do? It goes to the mRNA, all right, and joins, and here's the word you use often here, complementary codons. All right, so they come in, these three, they look for their right partners, they click in, all right, click, click, click. What are they carrying? Amino acid, amino acid, amino acid. I always refer to it as like a society. All right? So now all your amino acids are, you've clicked them in place. Click, 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 click. Your amino acids, what do? I join all of them. Peptide bond. What have I got? A protein. Okay. So let's put that in more biological language. There we go. Okay? According to the codons on the mRNA, there's your tRNA with the matching anticodons, complementary. All right. What do they bring? They bring the required amino acids to the ribosome. This is called translation. What are the amino acids? They're joined by a peptide bond, all right, to form a required protein. Okay. Now this is sometimes where this can get a bit tricky. The table below, you'll never need to know the amino acids. They will always give it to you. All right. It says the table below shows the amino acids that correspond with different DNA codes. Write down the correct sequence of amino acids. Write down the correct sequence of amino acids coded by the structure S. Let's go back to structure number S. Can you see here? This is structure number S. All right? And how do we group them? We group them in 3. 1, 2, 3. All right. So if we go back again, sorry. Oh, now I'm going forward. Sorry, yammer. If we go back again, all right, the table tells us that is the DNA code. All right? The DNA code. So let's go back here. We want the DNA code. A, what is it on DNA? A goes with T. U goes with A. G goes with C. Let's go back. Let me say forward. Okay, so I had a T, I had an A, and I had a C. I had a T, an A, and a C. So what's my very first amino acid? Methionine. All right, let's go back. My second amino acid, CCA. G, G, T, T, C, T. So I've got a GGT. Go back here. Where's my GGT? There we go. So this is number 1. This is number 2. This is number 3. There we go. All right. In order. If they're not in the correct sentence, the most important thing you need to remember, okay, is that you keep on forgetting which they gave you the code on the DNA, right? So you had to take your mRNA and you had to go back to the DNA. Right. Okay, guys, the next one is a diagram that you should become very familiar with, all right? When it comes to the human evolution section, you need to be able to recognize what this diagram is. Okay, so for starters, it is called a phylogenetic tree, right? And it's showing you the evolutionary relationship of the origin of humans. Okay, the dotted lines, now you need to read. The dotted lines here, you see all those dotted lines, they show us possible relationships, okay? Then we have these vertical lines over here. So that shows us relationships. Okay? These vertical lines over here show us time. All right. Which they were believed, how long they existed on Earth, so to speak. All right. So here, these little bars are time, and these little dots are relationships. Okay. Question number 1. Use the diagram to identify. Remember, you know, as soon as it gives you a number in capital letters, you can only give one. They are only going to mark the first one. Sorry. If you write 2 and the second one is correct, but your first one isn't, sorry for you. All right. You need to make sure your first answer is correct. That may have, now look at the question, use the diagram to identify one organism that may have competed. Competed means they must have been around at the same time, all right, as Homo heidelbergensis for resources. Let's go here. Here is Homo heidelbergensis. Who would have been around at the same time? You cannot use that one because look at their bars. I'm going to use the bars in pink. Look at the color of the bars. They weren't around the same time. All right? If you look at the bars, Neanderthalus wasn't around at the same time. But if I extend my bar, all right, who do I find would have been around at the same time, which obviously would have made sure that all right, there we go, Homo erectus. Have a look, take a pencil with you, right, because what you guys might have got a bit confused with is you might have looked at this one over here, and then you saw this one, and you saw that one, and you saw that one within the same time period. Don't, all right? They told you specifically the vertical bars, all right, showed you when they were in existence. And the only one that corresponds here would have been the Homo erectus. Okay, question number 2. It says identify the common, all right, the common ancestor that gave rise to Paranthropus and Homo. Let's go back. Okay. Where's our, here we go. Here's Paranthropus, and where's all our Homos? That's where the line, all my Homos came from. So if I go back, this one, common, there we go, they joined. Where do they both join? My Australopithecus afarensis. All right, my Australopithecus afarensis. There we go. All right. Now, the next one was a bit challenging because you have to think about it. It says, for what period of time did Australopithecus africanus, all right, exist on Earth? Show all workings. Let's go. Over here. So this is the bar. This is the bar that I'm looking for. How long did it live? Now, what I need to do is, this I know is 3 million years. But I now need to estimate how long that was. All right. So because it didn't live for those. So I'm minus, you could say maybe 2,4, 2,3 million years ago. What would my answer be? Approximately 6. Okay. Is it 6 million? All right, let's go over here. I'll show you. If we work it out. 3 million years, all right, minus 2,4, 0,6 million years. They gave you a bit of a time range here. I want to go back here. Have a look at the bar. In the beginning, they said to you, those bars are going to be important. So, there's my 3. Right. It couldn't have been half. It couldn't have been half, because here would have been 3,5. All right? In between, or 2,5, sorry. So you had to have judged 2,3, 2,4, all right? You'd done a nice, you could have used your calculator, or 3,0 minus 2,4, and you would got 0,6. Please, it's very important to put your units of measurement. If you do not, all right, put your units of measurement, you could well lose a mark. And maybe you get a mark just for putting the units, even though your sum was totally dear macaw. Okay, question number 6. Name one piece of evidence that we used. All right, when it comes to, all right, the human evolution, one piece of evidence that we usually used to prove that Australopithecus existed during the time period. All right, what do we use? Have a look here. Right, what could we also put here? Skull shapes. Instead of putting words, we could have put skull shapes. And when we have skull shapes, all right, how do we know the skull shapes? Because we got it from fossils. All right, we've got it from fossils. So when it comes to that evidence, obviously, okay, we're going to use, fossils as a source of evidence. Okay, now going back. Which organism, Ergaster or Neanderthalis, is more closely related, all right, to us? Let's go over here. All right, the question was, which one, Ergaster, have a look, all right, the organism Ergaster and Neanderthalis. Where's Ergaster? Over here. Let me go back. Ergaster. Oh, I'm not getting any of my pens. There we go. And Neanderthalis. All right, here we go. What do they have in common? All right, if you have a look, they have a common ancestor. I think we finished, Katlego. If I listen to Belinda whispering in my ear there. Are we ready? Are we finished?
Thank you very much, Cheryl.
It's a pleasure. Thank you. I'm exhausted now.
It's been a very, very interesting subject that we are doing here. It's DNA and genetics. Guys, remember to just keep, posted on our Facebook page. We're going to be throwing all of those links to our questions, exam question papers. You can get them on our website. Our website is learn.mindset.co.za, and that's where you can get all the revisions and the past lessons that we've had on the show. Thank you very much, Cheryl. It's been an awesome pleasure. Remember, guys, to be better than you were yesterday, and remember to keep, practicing, because practice really makes perfect. At Learn Extra on Facebook. I mean on Twitter and on Facebook, we are www.facebook.learnextra.co.za. From me, Katlego, it's really been an awesome pleasure being in your presence. You guys take care and all the best.